discussed about the mechanism of 2D page, two-dimensional polyacrylamide gel electrophoresis. Today we will discuss about the uses of 2D page. The 2D page is mainly used to study the changes in gene expression. The changes in gene expression means that the product or protein of a gene. The gene is made to synthesize messenger RNA and protein. That is the expression of gene. Every different gene produces a different type of protein, so that is the expression. If a gene is expressed, that means it will produce RNA, and the RNA will then translate it into protein. So that is the expression of gene. So that means a 2D page is used to study the changes in gene expression in an affected cells. What are the changes in terms of protein takes place in an affected cells can be studied by the 2D page. And by studying so, we can analyze the molecular basis of a disease. Molecular basis of a disease can be studied by studying the gene expression through 2D page. And that means that molecular, by analyzing the molecular basis of the disease, we can analyze or observe the effect of drug, toxin, and pathogens on a cell. How it can be analyzed, we can discuss by these two diagrams. So these are the diagrams of the plates gel obtained after the electrophoresis or page. Okay? So these are the bands. Suppose after a page we obtain a electrophoresis plate having these three protein bands in a normal cells. These are the extract of a normal cells, and the extract when done on a acrylamide gel, we obtain the bond bands. So suppose the protein bands of a normal cells we get, suppose these are the three protein bands obtained from a normal cell extract. Okay? So suppose this is the three bands A, B and C. That means when a normal cells is subjected for a study through 2D page, we get three bands. So this three bands tells the story about the protein of a normal cell. Okay? Suppose this is the affected cells of the same animal or the patient. Suppose this is the affected cells. We get the extract of affected cells and run it on a gel through 2D page. Excuse me. Okay, and in the affected cell we also get three bands A, B, but in the affected cells there is another additional band, that is D, which is not found in the normal cells band. That means that this is the additional protein or synthesized in affected cells subjected to any pathogens or drug or toxins or okay. And in these affected cells, this C is absent. That means that the gene which is responsible for the synthesis of this C protein is inactivated in these affected cells, and in the affected cells another gene is expressed or maybe it is mutated, that means कि another gene is expressed resulting in formation of another protein, that is D, which is responsible for the disease in the affected cells. By this method we can compare, by comparing the normal cells protein and the affected cells protein, we can analyze the expression of a particular genes by absence or presence of a protein. Okay, so by this method we can analyze the changes in gene expression of the normal cells and an affected cell by comparing the normal cells expression and the affected cells expression, gene expression. Okay, so this is the most important use of gene expression, and by this gene expression we can analyze the effects of drug, toxin and pathogens on a cell. Okay, similar way, okay, we can analyze the protein component of a cell at different stage of tissue development. When the development takes place during the organogenesis or tissue development, so on, there is a systematic expression, inhibition and expression of different genes. Some genes are active at a particular time, is inactivated at another time. Similarly, some genes are inactivated at a particular time, is activated in during the another stage of development. So by this way we can understand, we can analyze the what type of gene is expressed at a particular stage of development of a tissue or organ. Okay, by simply this way we can comes to a conclusion that this particular gene or this particular protein is present at this particular stage of development, and by this we can analyze or we can come to a conclusion what type of function a protein perform a particular stage of development. Okay, so by this method we can study the, analyze the component of a cell protein at different stage of tissue development. We can analyze or study the molecular basis of tissue development as well. Okay, and similar way, by this way we can analyze the response of a cell to a particular stimulus, and particular stimulus like hormones or cytokines. As we know, certain cytokines, particularly in the WBC activity, immune system's activity, so on, certain cytokines are important for their development, for their interaction. These cytokines activate the cells for the formation of certain type of protein. So that means when certain cytokines are, when a cell is subjected to a certain kind of hormones or cytokines, some new proteins are synthesized that will be studied. Okay, that means that will be studied affected cells. Okay, so by this way we can understand, we can analyze, we can study the effect of hormones or cytokines on a particular cells by analyzing the changes in the protein component of that cell which is subjected to certain external stimuli. Okay, similar way we can study the molecular basis of a type of cancer or Alzheimer's disease So what causes the Alzheimer disease or a particular type of cancer in a patient by comparing the proteins present in the blood of a person suffering from the disease, and by comparing that proteins component to a person which is having no disease or which is normal. So by comparing a normal person's protein profile, blood protein profile, and affected person's blood protein profile, we can observe or study the molecular basis of that disease in the affected person. So these are the uses of 2D page, okay, mainly by studying the changes in gene expression in the affected cells. So these are the uses of 2D page. Thank you very much.